Hello, my name is Dylan Rios, and today I'm going to be talking to you about meiosis. What is meiosis, and how is it different from mitosis? Well, first and foremost, mitosis involves the reproduction of somatic cells, while meiosis involves the reproduction of sex cells. Although these two forms of cell reproduction sound similar, they are very different in nature, though they do share some similarities. First off, we have interphase. But what is interphase? Well, interphase is a pre-meiosis phase that occurs in both mitosis and meiosis, and it takes up to 90% of the cell's life cycle. In interphase, the cell grows and replicates its DNA, and cell parts do everyday functions such as producing energy. Interphase is a vital part of a cell's life cycle, and without it, a cell would not be able to divide and reproduce.
If that was you on the phone and you on the bus, then who was flickering the lights?
Before we go any further, we must discuss the difference between haploid and diploid cells. Diploid cells are where there are two sets of chromosomes, one from each parent. Diploids are often linked to somatic cells. On the other hand, haploid cells are where there is only a single copy of each chromosome in a cell. Haploid cells will therefore have half the amount of chromosomes as a diploid cell would. Haploid cells are often linked to sex cells. This difference is important because it is one key way to tell the difference between sex cells and somatic cells. Gametes, otherwise known as sex cells, contain 23 chromosomes, while somatic cells contain 46 chromosomes. Next up, we have our first actual phase of meiosis, that being prophase 1. In prophase 1, centrioles are formed. Centrioles then produce spindle fibers, a string-like substance that attaches to the chromosomes and guides them to the opposite poles of the cell. But I'm getting ahead of myself. In prophase 1, spindle fibers do not actually attach to the chromosomes yet, for two main reasons. First of all, there is still a cell nucleus present. But what about when the cell nucleus disappears later in prophase 1, you may ask? Well, the second reason is because of the crossing over that occurs. During chromosomal crossover, two homologous chromosomes exchange segments of their chromatids between each other to produce heterozygous chromosomes. These heterozygous chromosomes contain DNA from two different chromosomes, leading to more genetic variation in the offspring. After prophase 1, we then have metaphase 1. During metaphase 1, chromosome pairs line up at the equator of the cell, and spindle fibers attach to each chromosome in preparation for anaphase 1. This is the main key difference between metaphase and mitosis and metaphase 1 and meiosis. Because chromosome pairs line up next to one another at the equator of the cell instead of single chromosomes, chromatids are not pulled apart from one another during anaphase 1. This is one of the main reasons why there are two sets of phases in meiosis instead of the usual one in mitosis, and is yet another key difference between meiosis and mitosis. The next phase of meiosis is anaphase 1. During anaphase 1, the spindle fibers spring into action and pull the heterozygous chromosomes to opposite poles of the cell. As I previously stated, full chromosomes are pulled to the opposite poles of the cell instead of chromatids. As you may be able to concur from that, this means that in order to complete cell division effectively, each stage must be doubled in meiosis. This means that at the end of meiosis, there will be 4 cells instead of 2, and therefore each cell will have half the number of chromosomes as somatic cells have. In this experiment, we simplify the number of chromosomes so that 2n equals 8. This number will change in later stages of meiosis. Once anaphase 1 is done, the cell can now move into telophase 1 and cytokinesis. Although I group these two phases together, they are not the same. During telophase 1, two new cell nuclei form, and cell cleavage starts to occur. During cytokinesis, however, the cell splits into two, and cytoplasmic DNA and organelles rush into the two new cells, and thus, half of meiosis is complete. You may notice that our previous number of 2n equals 8 has now changed to n equals 4. This is because each cell only has 4 haploid chromosomes instead of 2 diploid chromosomes. Next up, we have prophase 2. During prophase 2, just like prophase 1, Centrioles are formed that produce spindle fibers. The cell nuclei dissolves earlier than in prophase 1, as the cell nuclei is not present during the beginning of prophase 2. Other than this minor difference, there are pretty much no differences between prophase 2 and prophase 1. Next up, as you might have guessed, we have metaphase 2. During metaphase 2, chromosomes line up at the equator of the cell. It is important to note that unlike in metaphase 1, there are only 4 chromosomes per cell, and there is also no diploid pairs. This means that finally, during anaphase 2, chromatids will separate just like they do in mitosis. See, mitosis and meiosis are basically the same, give or take a couple of steps. As we begin to finish the long and strenuous process of meiosis, we have anaphase 2. This is starting to become pretty predictable, but in anaphase 2, spindle fibers pull chromatids to the opposite poles of the cell. The key difference between anaphase 1 and anaphase 2 is that in anaphase 1, diploid pairs are pulled apart, but the chromosomes stay intact. During anaphase 2, however, the haploid chromosomes have no buddy to pair up with, and therefore the chromatids must be pulled apart. But not least, we have telophase 2 and cytokinesis. During these final phases of meiosis, the new cell nuclei form and cleavage occurs, just like in telophase 1. Once the two cells split into 4, cytoplasmic DNA and organelles once more fill the new cells. The final chromosome count for each cell is 4. Thank you for listening to my presentation, and I hope you maybe learned a couple things today. Peace.